welcome grade 11 learners to another installment of human nutrition. In the last week we've looked at the overview of human nutrition. Today we will look at the processes involved in digestion. Essentially what we're looking at is what happens to food once ingested. How does that go through a process of being broken up? How is it absorbed and then assimilated into what the body needs? So those terminology is what we're going to look at today. So as we discussed last week, we looked at the overview of the digestive system. Just a quick recap on this. We're looking at the parts of the digestive system. What we should look at is, again, as we said, organisms that are autotrophs are able to synthesize their own food using process of photosynthesis. Here we're looking at animals that are unable to manufacture their own food, and we refer to them as the heterotrophs. All animals, including humans, are heterotrophic, and hence we say that they depend on the other organisms for their organic nutrients. Okay. Animals usually obtain their organic food in a complex, insoluble form, which needs to undergo a process of digestion. Essentially what we're looking at today is that process of how they become soluble so that they can be used at a cellular level. Before the animal tissue gets to use this food, it must be broken up into simple, soluble molecules. And that essentially is what we're looking at today, the process that is involved in converting those large, insoluble molecules into those soluble nutrients. Let's look at an overview of some important terms. We have discussed some of these. We're going to look at some of the newest terms that we haven't, or that will relate to the process. So we have looked at the digestive system. The parts that make that up would be the alimentary canal and the associated organs. We need to look at nutrition today in terms of the process in which food has been acquired and it undergoes that process of digestion. We discussed that homeostasis is a process that is involved in maintaining a constant internal environment, and that's more in the next episode when we look at how the body maintains a normality in terms of the levels of different organic compounds in the body. We looked at different modes of nutrition, and we're familiar with the terms herbivores, carnivores, and omnivores, and how their dentition varies based on their mode of nutrition. What we're going to focus on this week is the different molecules and how they're broken up. We all recollect from grade 11, we have organic polymers, and these are large molecules that then need to be broken up into smaller monomers, and that essentially are the molecules in their simplest forms, so the smaller molecules and how they're absorbed. When we look at them, we're going to look at the main building blocks in the body would be the monomers. So what happens to all the complex polymers, and how they are broken up into monomers and then absorbed. We discussed the process of peristalsis and how that propels the bolus along the esophagus into the rest of the alimentary canal. We're going to look at the importance of water in the process of hydrolyzing the organic compounds or the food to breaking them down. Mastication, I thought, would be an important word. It's an alternative word for the process of chewing, and that is essentially what occurs in the mouth when the food is chewed or it's broken up into smaller pieces, and for that the dentition is very important. And we find that herbivores tend to chew lots of their food, so the initial process of breaking down those complex food substances into smaller digestible parts where enzymes can work on is referred to as chewing. We looked at emulsification, how the liver releases bile salts, and that plays an important role in breaking down fat into smaller fat droplets. And then essentially what we're looking at today is the process of assimilation, which is a step in the process of nutrition. So an overview of the digestive system, we've discussed this, is that it contains two parts. It contains the alimentary canal and the accessory organs. An overview of the alimentary canal would be, contains the mouth and the mouth cavity, in which the tongue and the teeth play an important role in the initial process of carbohydrate digestion. We looked at the pharynx, and that's the opening that separates the esophagus from the trachea and how food then enters through. The esophagus, it's a muscular tube that runs at the back of the trachea, and that is essentially the part that carries the food from the mouth to the stomach. The stomach is it's the organ in which produces the gastric juices that play an important role in the digestion of the large polypeptides, breaking them down into smaller polypeptides. and then the small intestine and how the process of digestion occurs in there. We end off with the large intestine and the anus, which then wraps up the main parts of the alimentary canal. We also know that there are also other organs that are associated with the digestive system or the alimentary canal, and refer to these as the accessory organs. Here we're looking at the tongue, salivary glands that produce saliva, the pancreas that produces the hormones and the pancreatic juices, the liver that produces the bile, and how it plays its important role in the deamnification and the removal of waste products. And then eventually the gallbladder, which stores the bile for a little while. Okay, so having looked at that, let's go on to looking at some of the key concepts in the process of nutrition. So there are 5 main processes that we're looking at. Ingestion, and as I said, ingestion is the process in which food is taken in and enters the alimentary canal, and that is through the oral opening called the mouth, and there it undergoes a process of digestion. So the first process of digestion starts in the mouth, where we have mechanical digestion or the chewing of food, and that's essentially to break down the food. So the food is exposed to mechanical digestion and chemical process that changes the solid insoluble food into simple soluble substances. After digestion has occurred, we find that the food then needs to be absorbed to enter the blood or the body. So those end products of digestion are then absorbed into the bloodstream, and so throughout the digestive system, especially in the small intestine, there's a rich supply of blood vessels that actively take away the absorbed nutrients to the liver, where it undergoes further assimilation. Then we look at what happens after that. Those organic compounds that are broken up into monomers are then taken into the bloodstream. They reach a cellular level where the cells absorb the nutrients from the blood and use it to either build new structure or new compounds based on the requirements of the body for growth and for repair. And then finally, we're looking at the process of digestion, which essentially is the process that removes undigested and indigestible remains from the body in the form of feces. So guys, we've got to remember that whatever we eat is not necessarily whatever the body needs. So if you think of in terms of plant material, there is cellulose, which is undigestible. So we cannot digest cellulose. However, it does play an important role in terms of roughage, and hence the body cannot digest. So that is part of the roughage that is important in a balanced diet. Those undigestible substances are then removed from the body. We also have indigestible substances which cannot be digested. So we have substances that are not digested and that then are removed as waste products based on the body not needing them at all. So it's an accumulation of waste products and substances that have been utilized, and you have excess of that is then removed from the body. So guys, let's look at the key concepts in this. What is very important to understand is that the process of digestion is controlled essentially by enzymes that play a very important role. And as we recollect from grade 10, enzymes are affected by things like temperature and pH. So we need to consider that in the context of how enzymes function on the various organic compounds. So here, if you recollect from grade 10, we've got 3 main organic compounds. We've got your carbohydrates, which is starch, you've got your protein molecules, and you have your fat molecules. So we're going to look at how these 3 molecules, how these 3 main organic groups are broken up using enzymes into the monomers. And at this point, when these monomers are present, they will then undergo the process of absorption and assimilation at a cellular level. So when you look at your glucose molecules, plants produce starch, and those starch molecules are complex glucose molecules. So those starch molecules, and as you see, are complex. They will undergo a process of digestion or hydrolysis using enzymes which belong to the carbohydrates. So you got maltase, sucrase, fructase, galactase. So those enzymes will hydrolyze or will break down all your carbohydrates. So we refer to them as a group of carbohydrases that work on your carbohydrates, starting from glucose, fructose, galactose, maltose. And so if you remember your disaccharides, your polysaccharides, essentially that's what we'll be looking at. And then eventually all that is broken up into your glucose molecules or the monosaccharides, which will then be able to be absorbed into the cells and then used at a cellular level. The next group of organic compounds are your protein molecules. Again, going back to grade 10, remember that proteins are made up of amino acids. So The objective of protein digestion is to break down those polypeptide molecules into the simplest of the monomers called amino acids. Then those amino acids can be absorbed into the bloodstream. And for that, we're looking at a group of enzymes called the proteases. So the proteases work on your proteins, breaking them up into amino acids. And finally, we're looking at your fat molecules. And remember, if you recollect from grade 10, fat molecules are made up of 3 fatty acid molecules and 1 glycerol molecule. So essentially what this will happen is that these need to be broken up into your fatty acids and your glycerol. And for that we have a group of enzymes called the lipases. And these lipases will break down your complex fat molecules into the simpler molecules, which then can be transferred via the lymph vessels into the bloodstream. So guys, we've looked at an overview of the organic molecules and how they're broken up. Here's a table that summarizes the main aspects of chemical digestion in the body, and it's quite an important table for us to refer to in terms of where in the body, the name of the gland, the digestive juice that is produced, the contents that are released, and eventually the function of those contents. So when we look at the mouth, we've got to remember that the mouth is an important part that contains salivary glands, and these salivary glands play an important role in producing saliva. And saliva often has an enzyme called salivary amylase. Again, it belongs to the group of carbohydrases. So it's going to digest your carbohydrates, breaking them down into simpler, smaller carbohydrate molecules, or simpler organic compounds, such as your glucose. So the first process of carbohydrate digestion starts in the mouth, and that is through salivary glands producing an enzyme called salivary amylase. The next process or next part of digestion occurs in the stomach, and here you've got gastric juices which are released from gastric pits in the lining of the submucosa or the mucosa layer. Here you've got enzymes called proteases that are released. So you've got your proteases and hydrochloric acid, and that's the acid that is released to maintain the pH for the effective functioning of the protease enzymes. The function of the stomach is that it breaks down proteins and also provides an acidic medium and also can kill germs. So part of your body's defense mechanism is that whatever is ingested has to go through that acidic medium, and often it is a very effective mechanism in the destruction of most of your pathogenic substances that enter the body. Then we get onto the small intestine, in which the bulk of the digestion occurs. And here we see that the small intestine is supplied with enzymes, pancreatic juices from firstly the pancreas. It's also supplied with bile from the liver, and you've got your intestinal juices that come from the intestinal walls. So let's look at each of these separately. So what does the pancreas do? The pancreas provides sodium bicarbonate, and the function of sodium bicarbonate is to provide a basic medium. And if you guys recollect, from the stomach, it's an acidic medium that enters the duodenum, which is now containing a very alkaline or basic medium. And in order for those enzymes to function at that level, you need to have some mechanism to neutralize the acidic charge and convert that to an alkaline layer. And hence the sodium bicarbonate ions that are produced by the pancreas provide the basic medium for the effective functioning of the intestinal enzymes. You also find that your carbohydrases are released, and these break down your carbohydrates. You also have certain proteases that are released by the pancreas that breaks down your proteins, and you've got your lipases, which also will function on your lipids. So essentially from the pancreas you've got sodium bicarbonate, carbohydrases, and proteases being released. The next part that contributes to the small intestine will be the liver, and we discussed this. The liver produces bile, and bile contains bile salts, and bile salts play a very important role in firstly the emulsification of fats. That's breaking down the fats into smaller fat droplets. It also plays an important role as an antiseptic. These in terms of killing, destroying pathogens, having a sterilizing function, also plays an important role in the absorption of fatty acids and glycerol. So it provides an important function in terms of the absorption of fatty acids and glycerol. We know that the liver does absorb that through the hepatic portal vein being transporting those absorbed nutrients, and it contributes to providing a basic medium for the functioning of those gastric juices. The last part is obviously the intestinal walls, and these produce the three groups: your carbohydrates, your proteases, and your lipase. And again, your carbohydrates will break down your carbohydrates, proteases your proteins, your lipases break down your fats. So guys, we've looked at the overview of human nutrition and the processes involved in nutrition. Specifically, we looked at the enzymes and where they are produced, what their functions are, and what they break down in terms of how they work. Let's take all of that and let's apply that in some questions that we will look at in a short while after a little break. So have a little stretch break. When we get back, we're going to work through some questions. Welcome back, guys. So let's look at a few more questions on the process of digestion and nutrition. But I think let's have a recap of some questions so that we can get back into the parts of the digestive system, and then we'll look at some of the processes involved. So here we have an activity to do. It's a diagram of a human torso that has been labeled, that has been numbered. We've obviously got to label them and then answer some questions based on this. So here we've got that diagram labeled with various parts. So I'm going to very quickly go through the labels with you guys so that we can use these answers in the context of the question. So number one, guys, if you look at that tube that connects the pharynx or the mouth to the stomach is the esophagus. Esophagus that you should be able to identify very quickly. Number two shows you the liver. Number three shows you the structure here, which is labeled just below the liver, which will be the gallbladder. And we know that the gallbladder stores the bile for a little while there. And number four is the stomach, and that's the first part of the digestive system in which protein digestion occurs. Number five is the first part of the small intestine, that's the duodenum. Number six shows you the leaf-like structure, which is the pancreas, and that's essentially what you should be able to use it to identify. It's slightly below the stomach, and then obviously connected, if you look at it, it's connected directly to the small intestine via a little tube there. Okay, and then obviously number seven is the ascending colon, so that's part of the large intestine and it's going up, so and hence we refer to that as the ascending colon. Number eight is we're looking at as the cecum, and that's the first part of this large intestine. And number nine is the appendix, and if you look at the appendix, you'll find it's a little remnant or rudimentary or vestigial structure, appendix here, which is a remnant of a gland that used to produce an enzyme that was important in the digestion of complex protein molecules. Here we're looking at the descending colon, which is the last part of the colon. It's the descending colon. And number 11 would be the rectum, which temporarily stores the waste products, and number 12 would be the anus. So I'll very quickly label the diagram for you so it makes it a bit easier for you when you're actually looking at the label. So identify parts labeled, identify the labels for the following parts, and as I've done that for you, number one was the esophagus, number three the gallbladder, number five the duodenum, number six the pancreas, number seven we looked at was the jejunum, number nine the appendix, and number 11 the rectum. So very quickly I've done that for you. So let's get straight into some more questions relating to this diagram. Okay, so here, and this is quite an important question to look at. Make a neat label diagram of the cross section through part number 7. Part number 7 was the small intestine. So here you're required to draw a cross section through the small intestine. And guys, it's a very important part of the digestive system that we need to understand the structure of quite well. So guys, remember that the small intestine is a circular structure. If we take a cross section through it, you've got to show a layer with concentric rings, and I'm just trying to very quickly illustrate to you what it should look like. Here I've got a diagram and I'm going to go through that. So here you've got the outer serous membrane, you've got then the first layer on the outside is a layer of longitudinal muscles, Then you've got this layer here below the longitudinal muscles, which are your circular muscles, and collectively this is the muscular layer. Then you've got your submucosal layer here. Then you've got your mucosal layer, which produces the mucus, and you've got the Brunner's gland and the crypts of lubricant situated in there. And then finally the villi that are present on the internal surface, and that can be labeled as the internal lumen through which the food or the contents of the internal part. So what essentially I've taken here is that's the intestine. I've just taken this section and I've enlarged it there for you to be able to look at. Cool. So that's just a very quick, neat diagram that's labeled, and this diagram normally comes for about 8 marks, where you've got about 4 marks for the drawing and another 4 marks for the labeling of the various layers. More important is understanding the location of the longitudinal and circular muscles and the positioning of the mucosa and the submucosal labels, parts in your diagram. Let's look at more questions on this diagram. 1.3. Name the 3 pairs of salivary glands. Number 1.4. What are the muscle contractions that occur in part number 1? 1.5. What is the purpose of this type of muscle contraction mentioned in question 1.4? 1.6. Which digestive juice is secreted by part numbered 4? 1.7. Describe the pH and the type of food that is digested in part number 4. So guys, we've got 4 questions for you guys, or 5 questions for you guys to work through. I'll give you 3 minutes to work through these, and in between I'll flip back to the diagram so that you can engage with the diagram and the questions. So 3 minutes, get together, have a discussion, and when we get back we will look at your answers and my answers together. 3 minutes, guys. Welcome back, guys. So what do you guys think of those questions? Quite easy. Let's look at these solutions now. 1.3 was name the 3 pairs of salivary glands. So the salivary glands, remember that there are 3 pairs of them. You've got the parotid, you've got the submandibular, and you've got the sublingual. So there are 3 pairs of them that are situated around the jaw, so slightly below the jaw, on the inside, and then slightly above the jaw. So 3 pairs of them, and these collectively produce salivary amylase and the mucus that is required for mixing of the food. 1.4. What are the muscle contractions that occur in part number 1? And part number 1 was the esophagus, so let's look at it. So it's peristalsis, guys. So when we look at the contraction of the circular and radial muscles that propel the bolus down the esophagus, that process is called peristalsis. 1.5. What is the purpose of the type of muscle contraction mentioned in question 1.4? So we need to know what is the purpose of peristalsis. And as we discussed, it helps to move food through the alimentary canal and helps to mix the food with the other digestive juices in the small intestine and in the large intestine. So it helps to propel food along. It also helps to mix the food with the digestive juices, especially in the small intestine. 1.6. Which digestive juice is secreted by part number 4? And if you go back to the diagram, part number 4 was the stomach. So when we look at the stomach produces all the gastric juices and are responsible for polypeptide digestion. So your polypeptides or your proteins are digested by your gastric juices. So we're looking at protein digestion in the stomach. 1.7. Let's describe the pH and the type of food that is digested in part number 4. So we need to describe the pH and the type of food, which I've just done for you guys. So the pH of the stomach, guys, because it's involved with the digestion of proteins, the enzymes in the stomach, the proteases, all function within a very acidic medium. So the medium there is between 2 to 4. So it's an acidic medium that they function in. And what those gastric juices break down is proteins, and they break them down into your smaller polypeptides, which further undergo further digestion in the small intestine. Let's look at more questions based on, let's look at activity. This is another question looking at different questions obviously related to the digestive system. Study the diagram below and answer the questions that follow. So here we've got a closer up of just the liver. So as I would always do, I'm going to label this starting with number 1. So number 1 again, guys, is the stomach. Number 2 is this leaf-shaped structure called the pancreas. Number 3 is the pancreatic duct, and that's a tube that brings and drains pancreatic tube or duct that drains the contents of the pancreas into the small intestine. Number 4 is the hepatic portal vein. Number 4 is the bile duct. I'm just going to go through that. So that's the bile duct, and that brings basically the contents of the bile to the small intestine. Number 4 would be the duodenum, which is the first part of the small intestine. Number 6 is the gallbladder, and that's essentially what is going to store the bile. And number 7 is this large organ called the liver. It's quite synonymous with the triangular shape that you would see of it. So here we've labeled it. So you've got to identify the numbers to parts labeled 1 to 7. Discuss the 3 functions of organ numbered 7, which is the liver, and what is the function of part numbered 6. So let's look at these answers very quickly. So we've just done that together. We looked at the parts that were numbered. Number 1 was the stomach. We looked at that. Number 2 was the pancreas, leaf-shaped structure, the pancreatic duct. You got the common bile duct, the bile duct which brings in bile and meets with the pancreatic duct. So you've got that common bile duct that brings in bile that connects to the small intestine. So that's the common bile duct that we're looking at there. The first part of the small intestine we said was the duodenum, the gallbladder which sits below the liver, and obviously the liver. So quite easy questions to revise through. Let's discuss 3 functions of organ number 7. So guys, the liver, as we discussed in previous episodes, the liver plays an important role in 1, secreting bile. So it secretes bile that is stored in the gallbladder, and that bile is very important in the emulsification of fats. It is also able to convert glucose to glycogen, which is then stored in the liver and muscle cells for reserve energy. It also converts excess glucose to fat and stores it and keeps this as a reserve energy fuel. It plays an important role in the deaminification, or deaminates excess amino acids, resulting in the formation of urea, getting rid of waste products. And then finally, we said that it detoxifies harmful substances, preservatives, drugs, and medication that could be harmful to the body. So it basically will detoxify them and neutralize their harmful effects. 2.3. What is the function of part number 6? And if you recollect, guys, part number 6 was the gallbladder, and the gallbladder essentially is to store the bile temporarily so that that bile can be released into the small intestine when emulsification of fat is needed. So guys, we're going to move on to question 2, but I think you guys need a short break, and when we get back, we're going to look at question 2 in greater detail. So guys, let's get into the last segment. Let's look at some more questions, but we're going to get into greater detail of the processes involved and some of the enzymes and how they function. Let's continuing with that diagram. Organ number 2 is an exocrine gland as well as an endocrine gland. Now this is very important for us to unpack. So organ number 2 in this case is the pancreas, and that relates to that diagram that we were looking at before the break. I'm just going to very quickly flip back to that diagram so that we can recollect. So here's organ number 2, and that's the pancreas, slightly below the stomach, and it produces pancreatic juices which are released directly into the duodenum, but it also releases hormones, guys, and it produces hormones. So this is quite a unique gland in the body because it produces hormones and it produces gastric juices. So the gastric juices are released via a duct or a tube into the small intestine. However, the hormones are released directly into the bloodstream, and these are hormones such as insulin and glucagon, which play an important role in the regulation of blood sugar levels. So here we're seeing a gland that produces gastric juices and it produces hormones. Gastric juices being released directly via tubes into the small intestine, and the hormones being absorbed into the blood vessels that are surrounding the pancreas, and that's carried away. So essentially we need to look at why is the pancreas both an endocrine and exocrine gland. So what we should take from this is that the pancreas is both an endocrine gland and an exocrine gland, and that's based on its ability to produce pancreatic juices and release them via a tube or duct into the duodenum, as well as release hormones such as insulin and glucagon, which enter directly into the bloodstream. So let's read this question. Compare the differences in a table using the following points. So here we've got to tabulate, and guys, you've got to take note of the word table. 2.4.1, the concept of exocrine and endocrine glands, which we've just discussed. 2.4.2, the specific tissue that performs the exocrine and the endocrine function. So we've got to compare the tissues which perform the exocrine and endocrine function in the pancreas. 2.4, the substances secreted by these two types of glandular tissue. So we need to look at what the pancreas produces in terms of the endocrine function and the exocrine function, and where these respective secretions are targeting. So the target organs of the pancreatic juices and the hormones. And lastly, we've got to compare the methods by which the respective secretions are transported to their organs. So quite an interesting question. It's a comparison of the pancreas and the endocrine and exocrine functions. So let's see how to put these answers down in a solution. So again, the question did require that it needs to be a tabulation. So I've laid out my answer in a table. Let's look at 2.4.1. The first was, let's discuss the concept of the exocrine and the endocrine functions. Remember that the pancreas has an exocrine function, and it has glands which have their secretions released via tubes or ducts. So ducts are basically tubes. So you got the pancreatic duct that releases the secretions into the duodenum. However, the endocrine function of the pancreas is that it produces the hormones, and it's a ductless secretion. So essentially what we're saying is that these hormones are released directly into the bloodstream without any tube like the pancreatic duct. So here these are ductless glands that release their secretions directly into the bloodstream. 2.4.2, the specific tissue that performs the exocrine and the endocrine function. So what is the tissue that produces the exocrine function? So what is it that produces the pancreatic juices? So in the pancreas you have the pancreatic cells, and these pancreatic cells, guys, produce the pancreatic juices, your carbohydrates, your lipases, as well as your we looked at the other group, which would be your proteases. Okay, let's look at the endocrine function of the pancreas. So in the pancreas you've got patches of cells called the islets of Langerhans, and this should read islets of Langerhans, and these are alpha and beta cells that produce insulin and glucagon. So you've got your alpha and your beta cells that produce both the secretions of the endocrine function of the pancreas. 2.4.3, the substances secreted by these two types of glandular tissue. So what are the substances that are secreted by these two? When you looked at the pancreatic function, the exocrine function, they produce the pancreatic juices, and these are pancreatic juices that contain the digestive enzymes for those three respective groups. Let's look at the endocrine function again. So these are producing the hormones insulin and glucagon released by the alpha and the beta cells, which are then released directly into the bloodstream. 2.4. So guys, let me just say this: insulin and glucagon are hormones that are produced. Go back to 2.4. The target organ of these respective secretions. So what do these secretions target? When we look at the digestive juices, they go directly into the duodenum, and hence they will be responsible for digestion of your intestinal contents in the duodenum. However, when we look at insulin and the glucagon, these target the liver and your muscle cells, and will either convert excess glucose to glycogen, or in the case of glucagon, it will convert the stored glycogen back into glucose to bring the blood sugar levels back to normal. But more of that in next week's episode. 2.4.5, we're looking at the methods by which the respective secretions are transported to their target organ. So how are these secretions transported from the pancreas? So when we looked at the exocrine function, you've got the duct, which is called the pancreatic duct. So from the pancreas, you've got a tube that then secretes the secretions, releases the secretions into the duodenum. That is the pancreatic duct, and that is coming from the pancreas here to release the secretions directly into the duodenum. So this is called the pancreatic duct. What happens with insulin and glucagon? How are they released? These are released directly into the bloodstream, and hence they are transported via the bloodstream, and hence it's called the endocrine glands, and these, because of the ducts, are called the exocrine glands, or exocrine function of the pancreas. So what is unique about the pancreas is that it functions both as an endocrine gland as well as an exocrine gland, releasing their digestive juices, in this case pancreatic juices, directly into the pancreas. Quite an interesting question, but a very different way in terms of asking a simple question. Let's look at the next question. The following experiment was set up to test the effect of different pH values on the activity of protein digesting enzymes, which are normally found in the stomach of humans. Equal amounts of egg white, which is the protein, and solutions of different pH values were placed in each of the 4 test tubes. So here we've got digestive enzymes normally found in each one of them, and then these are placed on in test tubes with different pH values containing egg white. Let's look at the setup and let's see what questions are based on this. All the test tubes were kept in a water bath at a constant temperature. So here we're seeing a control variable in which all the test tubes were subject to the same conditions in terms of temperature. Let's look at test tubes A, B, and C. Here we're seeing 2 centimeters of enzymes into A, into B an enzyme, C enzyme, and in D boiled enzyme. And that is telling us quite an important concept of this experiment. In this, we've got to take note that the enzymes have been boiled, and that's quite important to understand going forward into the experiment. Let's look at the different pHs. So pH 2 was present for test tube A. Test tube B was kept in a pH of 7, so almost neutral. pH 8 was kept at a test tube C was kept at a pH 8, and test tube D was kept at pH 11. So here we're seeing an acidic, a neutral, a slightly alkaline, and then obviously a strongly alkaline or basic medium for this specific enzyme. What we need to remember is that this is an enzyme that is digesting proteins normally found in the stomach. So we've got to remember that an enzyme that works in the stomach prefers an acidic medium between pH 2 to 4. Here in this experiment, we've varied the pH and we've kept the enzyme at different pHs. So important to note that this would be something that would be the optimum pH in which the enzyme functions. So here we're having the optimum pH for which an enzyme that works in the stomach normally can function at. What you need to note here is that any enzyme that is boiled should denature an enzyme, and hence you're probably not going to expect any enzyme activity occurring in that test tube. Here we've got different pHs, which we will look at in terms of the context of what questions are based on this. So let's look at these questions. 3.1. If 2 centimeters cubed of enzyme was added to test tube A, how much of enzyme must be added to test tubes B and C? Give a reason for your answer. So we're going to discuss these questions after reading the first 3 questions. 3.2. In which test tube would you expect the egg white to be digested the fastest? Explain your answer. That's important. And 3.3. Briefly discuss the food test that you would conduct on the egg white solution to prove that it contained protein. So let's look at the solutions to these 3 questions on the next page. So, if 2 centimeters of enzyme was added to test tubes A and B, how much of enzyme must be added to test tubes B and C? Give a reason for your answer. So guys, when conducting any experiment, you've got to control the amount of substances that you add into all test tubes so that the variable that you're testing is not affected by changes in this case, the volume of enzyme that is being added. So I would, in my experiment, add 2 centimeters cubed of enzymes to each of test tubes A, B, and C, and D to ensure that I've controlled the volume and thus making my experiment fair and reliable, knowing that it's the volume of the enzyme that is constant and not the volume of the enzyme that is affecting the result. Remember that we are testing the effect of different pHs and not the concentration in this case. So the aim is to determine the effect of different pHs, and hence we've got to ensure that the volume of enzyme is consistent in all 3 test tubes. If we did that, it would change the aim of the experiment. 3.2 was, in which test tube would you expect the egg white to be digested fastest? Explain your answer. Okay, the answer to this would be test tube A. We've got to explain why. I'm just going to jump back to the diagram. When we look at test tube A, guys, test tube A contained 2 centimeters of enzymes. It contained pH 2, which is obviously the optimum pH at which your gastric enzymes function. So if we've got the gastric enzyme, in this case your proteases, at an optimum pH of 2, where would you think, in comparison to test tubes B, C, and D, would the enzyme function optimally? We know that pH 7 and 8 are far out of the range of the optimal, and hence these would have a significantly reduced rate of reaction. When we look at the last test tube, it has had the enzyme boiled, which means that it would be denatured, and you'll find that the enzyme has no activity in the last test tube. So essentially we can answer this question by looking at comparing it to the others. So in test tube A, we will find that the activity is the highest because pH 2 is an ideal pH for the enzyme which works on proteins on the stomach, which means that it's the optimum pH. Other test tubes have a pH ranges that will not allow the protein digesting enzyme to function optimally. D also has had its enzyme denatured, and hence you will find that there is no enzyme activity in test tube D. 3.3. Briefly discuss the food test that would be conducted on the egg white to prove that it contained protein. Now guys, this is again from grade 10 when we were testing the presence of organic compounds in different food groups. We had to conduct the protein test, we had to do a glucose test, we even did the starch test using iodine. And that you can recollect from even the previous episodes when we tested for starch in the leaf of a plant. Here we're looking at what test would we do to test for the presence of protein. And guys, recollect, it's the biuret test. And in the biuret test we had added the reagent which would be biuret reagent. And that reagent will turn the compound or the mixture blue. When we subject it to heat, it will change from blue intensity to a purple or lilac color. And that would be the positive test for the presence of protein. So we're going to use biuret reagent, and in the presence of protein, biuret reagent will turn from blue to a purple lilac color, confirming the presence of a protein. Let's look at the next question. So we've got a few more, and I hope that we can get through all three before the end of the session. At what temperature would you keep the water bath? Give a reason for your answer. Quite an interesting question. 3.5. How would your experiment be affected if you added few drops of concentrated sodium hydroxide to test tube A? Remember that sodium hydroxide is a base or it's an alkaline, so how would that affect the experiment? It will change the pH. We've got to explain that. And 3.6. Would you expect a positive test for protein in test tube D? Quite an interesting question. Let's get straight to the answers before the session ends. 3.4. At what temperature would you expect the water bath to be kept? And I would keep it between 30 to 40 degrees Celsius. And that's essentially because we're talking of enzymes in the gastric pouch or in the stomach. And that's the optimum temperature range which humans function at. So I'll keep it within the optimum temperature range of between 37 38 degrees Celsius so that we have optimum temperature for optimum enzyme activity. 3.5 basically asked you how would your experiment be affected if you added the drops of sodium hydroxide, and we've got to explain our answer. Knowing that sodium hydroxide is a strongly alkaline substance, it would affect the pH of test tube A, meaning that from pH 2, if you added a few drops of sodium hydroxide being a basic substance, it will alter the pH, changing it from 2 to probably 4 or 5, and this alters the optimum pH range. So this means that this will affect the pH range and it will slow down or even stop the enzyme activity from occurring. And that's essentially what happens with substances like sodium bicarbonate or an ENO that you take. So that's an effervescent antacid. So the idea behind that is that it neutralizes the gastric acidity so that the burning sensation is reduced. So essentially this would affect the gastric juices from functioning and affect enzyme activity. 3.6. Would you expect a positive test for protein in test tube D? And this is quite an interesting question because remember that in protein T the enzyme was indeed the enzyme in that was denatured, which essentially means that the enzyme cannot function. So if you test for the presence of proteins, would you get a result? Yes, the test for protein will be positive, essentially because the enzyme does not convert the protein, which is egg white in this case, to a different substance. So we will test positive for protein, and this is essentially because the enzyme that will digest the protein has been denatured, which renders it impossible to work. This means that the protein will still remain unaltered and the enzyme activity has stopped, and thus will still be present, and hence you'll get the positive test. That's the end of the episode, guys. Hang in, work smart, see you next week.